Hi, thanks so much for joining this video today. I'm going to be talking about the ChemiDocMP from Bio-Rad. This imager is a state-of-the-art, very sensitive, very highly reproducible, and very versatile imager that allows you to do all of your applications, from chemiluminescence to fluorescence detection to your DNA, RNA gels. It allows you to do your gel documentation as well as colorimetric, and our stain-free application, which can save you time and money in the lab. So it's an all-in-one, very easy to use, versatile system that our customers love. The software is very intuitive and the imager is very powerful. So I just wanted to give you kind of an overview of the system first. As you can see, we have our touch screen here. This also, you can have a mouse plugged in if you didn't want to actually use the touch screen to move the mouse around on the screen. As you can see, there's a USB port here. There are also 3 more USB ports in the back. So this can be used to take your images off of the imager, and you can also have this networked to a drive so that the images can be put there as well. So basically, there's a magnetic door that opens in the front, and the transilluminator tray slides out as so. What we have is, depending on your application, you'll be choosing a different tray to go on top of the pull-out tray. So this is our stain-free UV tray that comes with the ChemiDocMP, and this is for most of your applications that you'll be doing: fluorescence blots, chemiluminescence, your stain-free gels and blots, and ethidium bromide. So there's a couple different things. It tells you right on here the application types that you'll be using for each tray. And as you'll see later, the system won't let you choose the wrong tray for your application. It knows which tray it is by a little magnet here that slides into the groove here, so you can't use the wrong tray. So basically, it'll just slide right in there, and then you would close the door and take an image. We also have a blue tray. So the blue tray is for your nucleic acid applications. If you're doing any cyber staining, cyber safe, cyber green, your DNA, RNA gels, you'll be using the blue tray. And then we have a white tray. So the white tray is for mostly your Kamasi blue staining, your copper staining, zinc stain. So we have the variety of trays. And as you notice, there's little notches here on the tray, so it helps you to center your gel or your blot. And then you would simply fit it right in there, close this up, and close the door. And you can see on the screen live what's on the tray. And what's nice about this kind of door is that you always have the ability to reach your hand inside and kind of manipulate your gel and move it and turn it a little bit. Also with the screen, it's just like your phone where you can pinch and move and resize. Now I wanted to show you the touch screen a little bit closer. As you can see up here, we have a tab for camera and gallery. Camera is where you want to go when you want to take an image. Gallery is where you go to look at your images that have already been taken. Everything is automatically saved when you take an image, so you don't have to worry about saving any images. Now we just work down this screen here on the left-hand side. As you can see, we have the option for single or multi. So this instrument allows you to multiplex for your fluorescence blots, or you can choose single to take a single image. The next block down here is your image size. So there are preset small, medium, and large image sizes, as well as the ability to manually change the size. If you would like to, you just touch on it here and it will populate with a box to put in the numbers. And you can also, like I said, you can also use your fingers to adjust the size as well. Next here is your application. So here is where you choose which application you're doing. If you're doing nucleic acid gels, protein gels, or blots. And as you can see with the colors here, it tells you which tray you'll need for that application, whether it's a white tray, a blue tray, or the stain-free tray. So as you move your way down, here is your exposure. So this is where you can tell the imager how you want to take that picture. There is an auto setting and a manual setting. So the auto setting allows the imager to take an image, either optimal or rapid. So a rapid image is just going to take a quick and dirty picture, the best it can do, as fast as it can be to get a good image. The optimal is now saying, I want you to take as much time as you need to get the best picture that you possibly can. So the manual option allows you to go in and set a manual time. So you can choose this box here, type in 30 seconds, hit OK, and it'll do a 30-second exposure time. You also have the option for signal accumulation mode, which allows you to take a certain amount of images in a certain amount of time. And as that time goes on, the imager will take more and more pictures. The exposure gets longer and longer. And then when you go into your gallery, you can see each one of those consecutive images. You also have the ability to look at the binning and change those settings as well. So usually when I'm taking a first image, I like to just do the rapid auto exposure just to give the imager a chance to take a picture, and then I can determine from there whether I want to increase the exposure time or adjust any other settings. So then once you're ready to take a picture, all you would do is hit the green camera at the bottom. If you find at the bottom there is an exclamation point, that means that you have the wrong tray selected for your specific application that you want to do. So it won't allow you to take a picture if you're using the wrong tray, which is a great safety check. So as you can see, once the imager takes a picture, it takes you right into the gallery. It automatically saves your image so you don't have to worry about losing anything. In this view, you have the ability to use this transform button. So what this does is it populates these sliders, and all it's doing is changing how the image looks to your eyes. It's not changing any of the metadata or changing any of the actual image file. It's only how your eye sees it on the screen. You do have the option here to invert your image display. And you also always want to keep your highlighted saturated pixels highlighted there. And what that does is if you have any saturated pixels, you will see a red dot where those pixels are. So that can give you a warning if you are oversaturated. There's also a button here, auto. So if you want to just go back to the original way the image looked, you hit that button there. There's an image info button down here, and what this does is it populates all of your metadata. So you can go back to an image and look what the exposure time was and what your settings were. This is where you can also change what the file is called. So it pops up with the keyboard, you can change the name, hit OK, and it'll change the name of the file. Down here, you have some other options. If you're in your gallery, you can merge some images together. You also have the ability, here is the send save button. So if you do have a external hard drive plugged in, that'll populate here and you can save to that drive. You can also export as a publication TIFF, publication JPEG. You can also always want to export as ImageLab file, because that way this file type will be able to be pulled into our ImageLab software for further analysis. So this imager also allows you to set up users and have authentication so that nobody can go in and delete someone else's files that are in their gallery. So you do have the ability to set up an admin and have user logons. So that is pretty much an overview of what we have here. I'll show you quickly the multi. So what this does is it allows you to multiplex, and what you do is you just go through and choose which application you're doing and your different fluorescents. And then once you go to take a picture, it'll take each one sequentially, and then it'll also have an overlaid merged image of those files. And these colors, these red, green, and blue, are what we call pseudo colors. It's just random colors that we chose. You can always change these to be something else. So that's pretty much the overview of the online software, and we can go and show you the ImageLab software as well. But I just wanted to give you kind of an overview of that software and how easy it is to use. And thank you so much for joining this video.